Good evening friends. Welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about a plasmid cloning vector known as PUC19. PUC19 is one of a series of plasmid cloning vectors that was developed by Joachim Messing and his co-worker. Other vectors of the PUC series are PUC18, PUC9, PUC12 and PUC13. P stands for plasmid and UC is the abbreviation for University of California, where initial works on these vectors were conducted. PUC19 and PUC18 are very closely similar. The only difference between PUC19 and PUC18 is the orientation of the polylinker sequence or the multiple cloning site. So PUC19 is a plasmid cloning vector which is a circular double-stranded DNA molecule, and PUC19 is one of the most widely used vector molecule because when we use PUC19 as a cloning vector, then it becomes very easy to differentiate between recombinant and non-recombinant on the basis of differences of the color of the colonies that develop on a growth medium. This PUC19 is a circular double-stranded DNA molecule. It contains 2686 base pair. This vector contains an ampicillin resistance gene or BLA gene, the sequence of which has been changed so that it do not has a unique restriction site. In addition to this, PUC19 also contains a scoreable marker. This is the scoreable marker. The scoreable marker is the LAG-Z-alpha gene. This LAG-Z-alpha gene, it is a part of the LAG-Z gene of E. coli. And LAG-Z-alpha gene encodes N-terminal fragment of beta-galactosidase. And beta-galactosidase is an enzyme that catalyzes the breakdown of lactose to glucose and galactose. Within the LAG-Z-alpha gene, a polylinker sequence has been inserted. This is the polylinker sequence, which is also known as multiple cloning site. This polylinker sequence contains several restriction sites. Like other plasmid cloning vector, PUC19 also contains an origin of replication that derived from another plasmid vector known as PMB1. PUC19 is a high copy number plasmid. The transformed cell contains about 500 to 700 copies of PUC19. The high copy number of PUC19 is due to the lack of the ROG gene. And another reason behind this high copy number is a single point mutation that occurs in the origin of replication. PUC19 contains several restriction sites that are derived from the vector M13MP19. Friends, this is about PUC19. Please share, like and subscribe this video, and also please give your comments. Bye bye, good night.